Okay, so before we move on with the things that we started in the last class, which is basically epigenetics in 3 dimensions, I just wanted to show you one example of chromatin state identification through PCA followed by hidden outcome model. Now, since many of you are not interested in PCA, so the left side here, the 2 plots, you can ignore that. These are known as biplots, and they have superimposed the chromatin states onto this plot. So let's not talk about that. Let's only talk about the left panel. So what you see is the chromosomal position from in Drosophila genome, 16 kb to 17 kb, and this is in kb, so which means 16 MB to 17 MB. So this is 1 MB region, and each track here, each row represents the ChIP-6 signals of various chromatin associated proteins. And what you see here, let me take what you see over here. For example, these are the signals over here for proteins which are associated with heterochromatin, HP1, HP6, and so on. Okay, these are all activators over here, and likewise. Okay, so these tracks are activators. These are polycomb associated proteins over here, EZH1 and PG and others. Okay, these are polycomb associated proteins. Now, on what you see on the bottom here are the gene locations. Basically, each vertical bar is one exon, and if they are connected by horizontal line like this is one gene basically. Okay. And what you see here, so this is the chromatin type, which is inferred later on, but these are the three principal components, the leading principal components which were used to infer the chromatin state. So just look at this green state. So you see that the component 2 values are high while other 2 values are lower, right? So this is your heterochromatin then. So this is your heterochromatin. Okay, and the blue one is polycomb associated. So you have blue here, and you see that the component 3 values over here, component 3 values are high, while component 1 and 2 are lower, right? And red and green, red and yellow is the active states, and you see the activators are high in component 1. Okay, so this is what I was trying to explain on the other day. They just use 3 principal components to infer these states over here. Okay, so that was all. So let's talk about the transcription factor that we were discussing in the last class. So what we learned is that RNA pol2 are immobile foci inside the nucleus. And if you ask more precise definition of transcription factory, is the foci of at least 2 RNA pol2 molecules. Okay, it can have hundreds of those, but the minimal definition is to have at least 2 RNA pol2 molecules. And then we learned that in the inactive states, genes might be lying nearby only. Okay. However, upon induction, upon any kind of stimulus, transcription induction, these genes will move to the nearest transcription factory for activation. And generally, the transcription happens at the surface of the transcription factory. It's not inside. Okay, so it's not like pol2 is coming on top of the DNA and transcribing gene and rolling over the gene to transcribe. It's the other way around. It's the genic loci which are mobile and the RNA pol2 foci are immobile. So this was transcription factory thing, and somebody asked then how the specificity comes. So there is the concept of specialized transcriptional factories. The specialized one, how did they identify? The experiment which was done was to have genes. Okay, so they constructed genes. They had several genes, 7, 8 genes with same promoter under the influence of same promoter element. So they have this system in yeast. And when they looked at through microscopy, what they learn is that these genes are co-localized, okay, which means that in 3D they were co-localized in one locus for transcription inside the nucleus. So let's say this was the nucleus. So they co-localize for transcription, and then you do RNA fish, and you learn that all of them are actually transcribing at this site. And of course, when you're doing DNA, or this is DNA fish, if you're doing DNA or RNA fish, you need to have different colors for each promoter, right? Let's say this one was red. So you'll see a red dot out there, and another one is blue here, and another one is yellow. So not like that. Okay, in fact, you can make various different combination, and you can have 10, 12 colors in the same DNA or RNA fish experiment. So you can look at their co-localization. Then they learned that the similar promoters, which are bound by similar or the same transcription factory, they co-localize. How the transcription factories are localized at same place, somebody is asking. Well, there's something called phase separation I mentioned about, right? So phase separated condensates are stable units, stable configuration, which allow these transcription factories to exist. Okay, so this was specialized one. What is the other evidence that similar genes are coming together? As I mentioned, that there are methods to identify the proximity among genes. For example, you can have genome-wide method to find out what all genes are proximal. Let us say whether these two are proximal or these two are these. All of these are proximal or not. We have methods for that, right? Methods which identifies genome-wide proximity of genes or any DNA element, genome-wide spatial proximity. Okay, so once we have that proximity data and we know what all regions are spatially proximal, then you can look at their functions. So let us say I identified that these genes were spatially proximal, then I can go and see what were their functions, right? Were they enriched with particular function? Let's say the genes were associated with immunity, most of them, or any other thing, any other function, okay? So we can look for functional enrichment, right? Functional enrichment. And it turns out that similar genes with similar functions, they co-localize in 3D space. In fact, you can also infer this from protein-protein interaction, okay? Whether the protein products of these genes, let's say these genes in the end are making these protein products, okay? Whether they have interactions, whether the protein products had interactions, these were interacting with each other or not, right? So you can pull down the protein-protein interaction data and find out if the co-localized, spatially co-localized genes are also interacting at protein level. There are some papers which suggest that they do. However, uh, we in our own lab have studied that, but it doesn't seem to be a strong predictor of. Sorry about this dog barking outside. I'm out of station. I'm at home right now. I can't stop these dogs to come near my house. So, what is the stronger predictor of co-localization of genes? And of course, transcription is one thing. And many people say that the co-transcription of genes, the co-expression of genes could be a strong predictor, which means that if A and B, let's say this is A and B gene, and if I look at with respect to time, what is the expression of A and B gene, and if they are co-transcribing, then I could say that this is the way that they are co-transcribed. So whether the co-expression of genes predicts the co-localization of genes in this space, many people have shown that. However, it turns out, just give me a minute, I need to shut down the door. Yeah. So, however, I mean, different people have their different conclusions, and it turns out that I'm not trying to advertise my own work, but it turns out that one of the PhD students in our lab found that it's actually co-replication of loci, which is a strong predictor of co-localization. Why? Because if you look at the co-localization, you are not constrained by transcription factories, isn't it? If I look at the co-localization of genes, it will give me all kind of co-localization that could also be associated with DNA polymerase or transcription, right? So if you identify the co-localization of genes in an unbiased manner, then transcription factors could be one part of it. Okay. However, the majority of co-localizations can be explained by the co-replication of the engaged loci. Okay, nevertheless, transcription factors do exist, and then not only transcription factors, we also have replication factors, right? So we have transcription factors. We also have replication factors. It turns out that DNA repair foci are also limited. The genes move to certain limited foci where DNA repair is happening. Okay, splicing also happens at sites of splicing, or what we call as nuclear speckles. These are the sites of splicing factors. Now, it has to be convincingly shown that splicing indeed happens for most of the RNAs at the nuclear speckle sites. They are the storage of splicing factor, it is known, but nobody has shown that splicing is actually happening inside it. Some people have tried to introduce, uh, when they introduce intronic RNA, okay, in a gene which was not having intron, it tends to co-localize with nuclear speckles. But whether we have a same single nuclear speckle for multiple genes, okay, we do not know. Or there are evidence out there. There are several lines of evidence that I am not aware of in other ways. Okay, so these are few examples of uh, subnuclear localization, subnuclear compartments. Okay, there are many others, right? So you might know some of them already. For example, PML bodies. Have you heard of these? This is promyelocytic leukemia bodies. This is the name of a gene, PML, PML1, PML2, PML3, likewise. We have, so should I elaborate a bit on PML bodies, or you are aware of it? So, okay, so PML bodies are, so largely the size will be around 0.5 to 1 micron, and there can be variable number of PML bodies inside the nucleus. So the PML bodies are actually the site of simulations. Okay, a lot of PML bodies come, they come together. Okay, so again the phase separation thing, they are coming together, and they actually make a hollow-like structure inside where you can have other proteins coming inside. For example, you can have other proteins coming inside. Okay, and proteins get post-translational modification here. For example, simulation. The PML bodies themselves, PML protein themselves get simulated, and they also simulate the proteins inside. Right. So let me complete this cycle here. The green color is your PML proteins, and the red color are the other proteins. And so simulation is one thing, simulation. And there are other modifications also. Ubiquitination also happens inside. Simulation. You have ubiquitination. You also have acetylation happening inside. And likewise. Okay, so one of the interesting properties of PML bodies is that they respond to interferons, so which suggested that they may have a link with the viral infection. In fact, the viral DNA and proteins, viral DNA, RNA, and proteins, they co-localize with PML bodies. Whether they use PML bodies for their function or whether PML bodies have any role in immune system and so on is not entirely known. Okay, so primarily most of the PML bodies function. They seems to have a role in apoptosis, transcriptional regulation, DNA repair. Let me write it down. DNA repair seems to be regulated by the post-transcriptional modification of the involved proteins. Transcriptional regulation and apoptosis. These functions are regulated by indirectly by these modifications of the proteins involved in these functions. Okay, so they co-localize here. One of the interesting protein which co-localize here is the P53. It gets phosphorylated inside these bodies. Okay, so that's another example. Another example is causal bodies or coilin bodies. Coilin is the name of the protein which makes these bodies. Now, what do they do? So again, the size would be around 0.8 to 1 micron in size. So they also vary in numbers, cell to cell, and in which cell cycle stage the cell is. Okay, so they assemble, disassemble, and so on. They also respond to environment, like the one here, PML bodies. They respond to viral infection, either assemble or disassemble in some condition. They also disassemble. They are generally formed upon stress, some kind of cellular stress. Okay, so they are very dynamic structures. There's still hot debates on their function. Causal bodies now, they are the sites of, if I recall correctly, sites of maturation, sites of URNPs maturation. Maturation means again the post-translational modifications. One of the post-translational modifications is 2' O ribose methylation of RNAs in URNP. So you know what the URNA or URNP, these are the proteins involved in splicing. These RNAs are involved in splicing of other genes. Okay, so these are the maturation sites of, so let us say inside the nucleus, you have these sites somewhere here. Let's say this is the causal body. So maturation will happen there, and then we have nuclear speckles where the splicing is supposed to happen. So, or the uRNP is supposed to localize, okay, with the pre-mRNAs. So these uRNPs, they are transported from causal bodies to nuclear speckles. So this is your causal body, and this is your nuclear speckle, and then the splicing happens over there. So this is the role of causal bodies. You already know another condensate, and that's the heterochromatin thing, right? Or let's say there's another popular one, and that's the nucleolus, right? So I don't think I need to introduce you the function. So this is the sites of ribosomal biogenesis and the rRNA synthesis, right? You have nuclear speckles, So interest factors are the, we can write over here. It has other functions also, and later on we will learn. Nucleus factors. So they are the storage of, so splicing factors localized there. Splicing factor SC35 and likewise the other splicing factors localized there. Some people have shown that they are also the site of active transcription. Somewhat they overlap the function of transcription factories. Right. So likewise, you have heterochromatin, which makes large blobs, which are generally peripheral. They are generally peripheral to the nucleus. They would localize like that. Heterochromatin blobs. Okay, this reminds me another thing, and that is this is happening. This will happen in most of the, let's say in human, this is the case. Diurnal mammal, and let us say this cell is your rod cell in your retina. Okay. So interestingly, the same thing as inverted configuration. Right now is peripheral, right? So this has inverted configuration where the central part is heterochromatin in nocturnal mammals. And again, the rod cells. Now, can anybody suggest me what's happening here? Why is this so? I'm only talking about one kind of cells, that is the rod cells. You might wonder what animals am I talking about? So you can think of mouse here, rat here, maybe rabbit too, and you have human. Now you might say, well, you are talking about primates on one end and the rodents on the other end. That's not the case. In fact, we can draw a phylogenetic tree to clarify this. So let us say your mouse over here. Human or monkey, which are diurnal, and then am I connected? Sorry, I can't hear you.
Your screen is not visible.
Yeah, it disappeared suddenly. I don't know what has happened. Just give me a minute. Sorry about that. Sometimes all kind of magics happen. I hope you can see this now.
Yes.
Yeah. So let's say human or monkey. And let's say some carnivore over here, cat, and maybe a perissodactyl or artiodactyl cow or horse, or maybe pig. Right. So now we have a fair bit of nocturnal and diurnal animals, isn't it? So what all are nocturnal and diurnal here? Anyone? So this guy is nocturnal, right? Nocturnal. Lemur is a primate, nocturnal primate. And then we have, this is diurnal, right? This is cat is considered as nocturnal. Okay. Now, if we look at the organization of chromatin in each of the species, the rod cell of each of the species, let's say this is the nucleus. It turns out that this is central localization of heterochromatin, central one, peripheral one, central one. So it's not only mouse rats, so most of the nocturnal animals will have more central location of heterochromatin. Now what does it has to do with the nocturnal habitat? What is the speculation? That's the configuration of a diurnal and nocturnal animal. Any guesses?
I think that you see behind so those kind of the genes are also recognized because they regularly see not in the light but in the night.
So you're trying to say that different kind of genes expressed,
Yeah, maybe
but
that.
that doesn't need it to be completely reversed. Right now, it's completely inverted, right? And in fact, people call it as inverted rod nuclei. Well, it turned out that now this is right now. Uh, you may say that this is at hypothesis level, but there is some evidence. Okay, so computer simulation as well as some experimental work. Okay, let's say I have a usual peripheral heterochromatin here and an inverted one over here. This is inverted one. So when I say peripheral, that would also mean that sometimes you also have mixed pattern. Okay, just to tell you, sometimes you have intermediate state also like that. Let us consider that this is peripheral. And what I'm going to plot is the intensity of light transmitted from these nuclei. Okay, let's say there is a light going from this side. And what I'm going to plot here is the intensity of light, of transmitted light. And people learn that this gives you more chaotic transmission of light, right? However, this one will give us more focused light. Why is that so? When are you able to focus the light? Fundamental physics. When you have a concave convex lens, right? What it does, this heterochromatin, this is somehow is focusing the light, okay, while this one is not able to focus the light and you get the chaotic pattern. Right now, you might wonder that we don't have one such nuclei in eye, right? You have multiple such nuclei. Each one does it. Each one in its own capacity. It will focus, try to focus the light. And since the nocturnal animals have very little light available in the night, so they have to see. So they need more. They need more focusing of light, right, to visualize in the dark. So now what you see here is some kind of moonlighting of heterochromatin function. Moonlighting means when you have another function apart from the known function. For example, if you are studying, so you are a student, but if you also drive a taxi in the night or work in a restaurant, that's called moonlighting function, right? So heterochromatin function is to silence the genes stably, right? What we see here, it's also helping to focus the light on the retina. Okay, so this is another function of heterochromatin in broad cells. It may turn out in some point of time that there are series of papers which refutes this claim, but as of now, this is the understanding. And the authors who have done this, I will upload this paper. They actually say that they hypothesize based on their simulations and observations so far, and nobody has actually challenged this view for the time being. Okay, so you see many interesting things are happening inside the nucleus through these condensates, right? Now, how are they made, right? So they are made through the process of phase separation, liquid-liquid phase separation that you hear. Was it taught to you in any of the biophysics class, biochemistry class? Anyone? I couldn't hear.
No, sir. Yeah, I mean, it was not taught.
Actually, whenever I, this is interesting,
No,
when
sir.
I ask about anything, even if I ask you, was PCR taught to you, half of the class will say no. Okay, so anyway, so I'm not the right person to talk in detail on phase separation. This is largely the physics domain, sorry, biophysics domain or chemistry. So what happens is that how is that driven? Phase separation. Of course, there are thermodynamic forces, right? And I will give you the example of entropic force. Okay. And there's an interesting property that you need for phase separation, that is multivalent interactions. And we will see what are those multivalent interactions in a while. Okay, the entropic thing. So I'll try to explain you based on what I know. Okay. So let us assume that there are two molecules like that. You can consider these as two RNA Pol 2 molecules. Okay, and then you have several small molecules. Let's say this is one system, okay, that we are looking into. So then there will be some volume around these bigger spheres which will be inaccessible to the smaller molecules, and I'll tell you why. It's simple. This is the not the whole of it, but the center of the smaller molecule. This is our radius of this small molecule, right? So the center of the small molecule or sphere cannot go inside this dotted line, right? That's understood. So this is generally called as excluded volume. Okay. Now, let me tell you what happens, what can happen in one scenario. Let us say these two larger molecules come together. So what's happening? There is some depletion of excluded volume now. What is that depletion? This depletion, right? So excluded volume has decreased now. There was a volume which was inaccessible to smaller molecules. That volume is decreased now, which means that more volume is available to smaller molecules to roam around, right? If that happens, what will happen to the entropy for these smaller molecules?
It will increase.
Yeah. So delta S is going to increase. Delta S was relatively, so this is relatively lower here. So which means that this process would be favored, right? This is called, this is depleted volume now. Depleted volume. So this force is called depletion attraction. Depletion attraction. Very underappreciated phenomena in thermodynamics. Okay, not many people talk about this. Now you might say that, well, what about the interaction of smaller molecule with the larger molecule? Let us say if the interaction strength of the two different molecules is greater than the interaction strength of the two larger molecules, then the first scenario will be favored, right? Isn't it? So, which means that there has to be a condition that needs to be satisfied for phase separation to happen. For example, let me take two molecules in two different colors. This is the mixed state, let's say, of two molecules, right? It is not phase separated. You can have a phase separated scenario, which would look like that, two of them are phase separated, all red are together. You can consider one of these is the larger one in the earlier diagram, and the other one, right? That's your phase separation. So this can only happen if the uh, energy or entropy of homotypic interaction is lower than the entropy of or the free energy of heterotypic interaction, right? So the condition that needs to be satisfied is the following. The energy of heterotypic should be significantly greater than energy of homotypic interaction. Homotypic means the interaction of the similar molecules, right? So sometimes this is given in an equation which people write in this way. O minus E homo should be divided by kT. kT is the thermal energy, right, at room temperature. So it tells you that how much energy would increase by 1 unit of temperature increase. Now, how much temperature is needed to increase the energy, thermal energy by K unit, which is the Boltzmann constant, to be very precise. And if this quantity is significantly greater than 1, then you have phase separation. Okay, so focus on this equation. Should be understood to layman, right? So that will lead to phase separation. So that also explains you that although many molecules phase separate, but then there are many other molecules which do not phase separate. Why? Because this condition is not satisfied. Okay, so this is one thing, and you would have seen this. This can be considered as interaction strength, basically, given by some parameter. You can name anything. Anything, let's say. So I think you would have seen this diagram of interaction strength versus concentration, right? Or the way around, concentration of, let's say, proteins versus interaction strength, inverse of interaction strength, which is nothing but temperature. If you increase the temperature, you will mix the things. Interaction strength will be weaker, right? So this is inverse of interaction strength. Concentration means volume fraction. So volume fraction has a precise meaning which tells you that the volume of one of the molecule, okay, with respect to total volume. If it is less or more, it means phase separation, right? If it is around 1, which means that the volume is like 50-50 for both the phases, okay, or 0.5, you could say, then it means there is no phase separation, there is a complete mixing. And you would have seen this diagram coming in at many places, this kind of phase separation diagram. It tells you that you have mixing over here, mixing of two molecules over here. While below this you have demixing. Then this is called as your critical temperature. This is critical temperature, TC. And if it goes somewhat like that, it becomes a typical phase separation diagram from liquid to solid. If this goes like that, right, then this is critical temperature, and above that there's one phase, and below that there's another phase. So for these things, I think you need to refer more of thermodynamics of phase separation. You need to refer to your biophysics classes. This is not the ideal course for that, but I think this should be understood. It should be straightforward. So this volume fraction means volume of one particular molecule, right, over sum of all volume, right, total volume. This is V fraction. Okay, so phase separation occurs, and when you have certain interaction strength, which means that if you your interaction strength is stronger, so interaction strength, if I plot, it becomes like that. Interaction strength is stronger here. Then you have phase separation, but when you increase temperature, the demixing happens. What I wanted to talk about was, okay, so I think if you understood this, you understand what are the entropic forces that are working in phase separation, and this one. The second thing was the multivalent interaction. What is that? So multivalent interaction is, assume that there is one molecule like that, and they have only one interaction surface. Okay, then they will interact, right? And they will end up having a scenario like that. Just the dimers will be floating around. Why? Because you just have a univalent interaction. Just there is only one binding site out there. There is one configuration which is needed for their interaction. However, assume that you have multiple such things, multiple such sites. You have it over here. And now if you reach a critical temperature and concentration, this is going to make a condensate like that, right? Well, these are the binding regions. Right? So this is your, this is the essential requirement. This is one of the essential requirement for phase separation. This is one of the reasons the disordered proteins, which would look like this. Some part will be ordered and other is disordered. And this disorder gives extended surface for multimeric interactions, and this could lead to phase separation. In fact, HP1 has a disordered domain. This is heterochromatin protein 1, and it has been shown to phase separate, to make heterochromatin phase separate through disordered domain, through intrinsically disordered region IDR. Okay, Professor Samrat Mukhopadhyay works on IDRs and their phase separation. So in his classes, you can ask about. In fact, he must be teaching phase separation in classes in great detail. But this is the phenomena underlying these condensates that we see inside the nucleus, not only inside the nucleus, but within the cytoplasm also. And this doesn't mean that all the proteins phase separate into this homotypic complexes. There are proteins, if you label them, there are proteins, if you label them, so this is the nucleus and you label that protein in green color, you will just see homogeneous spots all over the nucleus. Very, very homogeneous that you can't even see dots, you just see whole thing is just a smear. No condensate at all. Okay. So that would mean that the conditions were not favorable. Conditions like these were not favorable for those proteins. Okay, that was one of the things about the transcription factories. Let us come back to the process of transcription now. So let us say that we want to identify. I said that we can identify all the genes or the regulatory elements which are spatially proximal to each other. How do we do that? So there is something called proximity ligation. Get the concept of proximity ligation. Does anybody know about this? This is also used in protein interactions. Well, it simply says that something which is proximal, you ligate them. Okay. Proximity ligation based assays. Okay, we do these assays to identify the regions which are spatially proximal. So it's already 5:45. So should I continue or should we discuss this topic in the next class? Because this is again a separate topic now. I introduce you that genes do come together. Now we are going to discuss the techniques to identify those interactions, and then we will we'll see what did we learn from these techniques. What is the present stand? What do we learn from these techniques, which is advancing our understanding of molecular basis of transcription, replication, and so on. Anything which is proximal, let's say these two DNA fragments, you ligate them. Okay, that's what it means, proximity ligation. And if at all this DNA fragment over here was conjugated with the antibody over here, then you can identify protein-protein interaction because they were spatially proximal. That's why they came together and we were able to ligate the conjugated DNA fragments. So let's discuss this in great detail because it's not one technique. There is a whole industry of techniques now. Generally, originally started by one fellow, Job Decker, and we'll see what are this family of proximity ligation based assays. And one is dependent on another. Okay, if you learn the one technique, the second one is actually logical expansion of the first one, and third one is the further extension of same concept. So it has to be taught in one class. So let's meet in the next class to discuss this. So there is a question. Okay, somebody said next class. Okay, any question? We have time, 5 minutes for questions.
Sir?
Yes.
So we have discussed, I mean, we know basically about these modifications like methylation, acetylation and all, but we haven't been taught yet about the simulation. So in any of the class, could you touch upon briefly that simulation?
Well, simulation is another post-translational modification of proteins. And this is not well studied in terms of chromatin regulation. Okay, so these are not the modifications studied for histone. At the most, there would be some assay which is genome-wide, might be phosphorylation. Okay, most of the modification which are associated with chromatin is the acetylation and methylation. Some people have studied ubiquitination, perhaps. Okay, so if there is a link with the chromatin, I will try to explore that and highlight in one of the class. This is another important modification. So this is not actually epigenetics that we are talking about. This should have, I mean, ideally, in ideal scenario, this should be learned in a course on cellular biology or whatever. There's something called PARPylation, which is poly-ADP ribosylation. I might have misspelled it, but this has something to do with something called PARP proteins. Poly-ADP ribosylation. These are very important. This is very important modification. Some of the proteins actually have their peculiar function, chromatin-associated function, because of PARP. For example, CTCF. Okay, is when the CTCF is PARPylated, then it has a particular function of insulation. Otherwise, it has some other function. Okay, so some functions are, now this is not extensively studied. There are only few groups who have shown that for this particular and insulation function, you need to have PARPylated CTCF. Otherwise, this function is compromised. Okay, I will see. We can talk about this relatively less studied function in one of the class. But again, since these are not well studied, I will not go in detail. They are not part of genome-wide studies, genome-wide assays. So what generally people do, the way they study this modification is they mutate or knock out the gene which is important. For example, for phosphorylation, you have kinase and all that. The PARP enzyme, you remove it and see what's happening at your locus or genome-wide. So that kind of studies will be around, knockout experiments. Not really the assay, but if there are assays which are done recently and giving a new insight, I will highlight that in the next class. Was there any such thing that I promised in the last class also that I will look into? If I'm missing something, keep me reminding. So I'm writing this down. PARP simulation. Ubiquitination, was it taught? Because there is a research group in ISOMORI who is working on ubiquitination. It's important for protein degradation. right? Ubiquitination of proteins.
Yes, sir.
Ubiquitin makes a tunnel-like structure, and you need a disordered region to go inside of the target protein, and it degrades it. Okay, so ubiquitination has been taught. So I will see if I have some details on simulation, phosphorylation, compactylation. Okay, okay, I will give you some information on that. So there are more aspects of 3D genomics, but once we learn about this technique of proximity ligation, we will see, okay, this is the way it is being done, and then we will discuss what did we learn through the result of this technique. Now, ideally, this should be part of experimental techniques of epigenomics in the syllabus. But if I do not teach this, then it will be difficult to appreciate how the present understanding of chromatin regulation is achieved. So it is worth acknowledging the authors of these techniques. So let me elaborate on this, and whatever techniques will be left out will be taught later on. And those techniques are the techniques to study DNA methylation, techniques to study open chromatin, DNA footprinting assays, FAIR-seq, which is again for open chromatin, but there are different assays, DNAs, seq assays, and so on. Okay, any other questions? Techniques used for epigenetic studies. Yeah, so I said about chip thing, but I will cover all of them. I hope chip is not a problem to. Okay, let me just give you the small glimpse. For example, what you do in chip, let's say you have uh, this is your protein of interest X, which is binding at multiple places on DNA, right? Or transcription factor X. What you do is you fix it first with formaldehyde. You will see this step is coming again and again, the formaldehyde fixing, and I'll tell you in the next class what do I mean by it. Okay, you fix the things over there, and then you chop it through sonication, right? This is a one fragment, and this is another fragment now. Sonication or restriction enzyme. So then you use antibody to pull down your factors, primary and secondary antibody, and then all the noise. The noise is rest of the DNA which is not bound with your protein. This is rest of the DNA, okay, which is bound by some other Y protein, Z protein that you're not interested in. So in the end, through this assay, you will have new DNA fragment bound with your protein of interest X, right? Then you remove the protein thing and isolate DNA, and then you got the set of DNA which were bound in situ by your protein of interest. And once you have this, you can sequence them. How do you sequence? You do next generation sequencing of this. You add adapters, universal adapters, and they are different for two different ends. Okay. And then you do next generation sequencing. You will know how much each fragment is present, right? Each fragment might be one promoter or enhancer region. And then you plot this across chromosome, and you will find these peaks. Peaks means more copies amplified of this region, right? So more copies over here, less copy here, more copies again here. There will be peak out there, right? So in NGS, you also do amplification before NGS, right? So that you have detectable amount of DNA to NGS. So this will be repeated later on again, but since ChIP-seq word is coming again and again, I just gave you the brief introduction. Later on, we will combine all immunoprecipitation based chromatin techniques in one of the class. However, in the next class onwards, proximity ligation would be covered in detail. Okay, because it's important to know, this is a whole class of techniques. It's important to know how were they innovated, what was the original objective, and what are we studying now, and where this is leading us to. Okay, there's no more question. I can end the class now. Any more question? Okay, that was all for the day then.
Recording